we'll be discussing, it's in the wrong order, but we'll be discussing standards for these quantitative experiments and then how to set up calibration curves. And this is one larger slide deck, and the last half of it is a step-by-step walkthrough on some of the things that you would do in Skyline to build a calibration data analysis processing method. So, okay. So I had sent an email last night. This is a link from the study I discussed earlier this morning, where it's looking at fully Skyline processed or integrated data files from the experiment that has light C13 N15 synthetic peptides and fully labeled N15 peptides present in the sample. And you can download either of them, but the examples that I am working from will be the post-DS. DS is for desalt, so peptides were added in post-desalt in that experiment. So, how many of you set up quantitative targeted methods and do not use standards? It's okay, don't be shy. All right, why not? Haven't optimized, haven't like selected what you want to go for? Same?
They're in my desk.
Okay. So for some, it's a matter of availability. For some, it's cost. So what I'll go through are some alternative options in standards if you can't afford or you don't have access to an isotopically labeled exact form of your analytes, and then different ways we can use them. And so in addition, I have a little bit on internal standards versus external standards, and then for targeted peptide experiments, starting with peptides, extended peptides, protein standards, or a global proteome standard, such as something like SILAC, and then where you can get them and where you can use them. So again, just to reiterate this, why use standards? So inherently, mass spectrometry is not very reproducible, and it's not absolutely quantitative. If you take a sample and you measure the raw peak area of a target on one day, and then that same sample 2 days later or 2 minutes later, you may not get the same absolute response. And that's because there are a lot of variables affecting the generation of those ions and the detection of those ions in the instrument. So having a standard is always a great idea, because you can always quantify, you can make sure, you can qualify your standard, and you can always compare the two. So again, going back to the comparison against Beer's law, we would always expect that if we see an increase in our MS signal, that correlates with an increase in our concentration of our analyte, because they're on opposite sides of the equation. But the background signal contributes to our overall signal, and then there's this coefficient that we cannot calculate ahead of time or theoretically predict at this point. And because these are all unknown, the best way to start is to have a standard. So they account for variability, and depending where you put them in your workflow, if you put them in early on or you have an isotopically labeled proteome, then they can account for sample preparation, reduction, alkylation. Things that you put in even at the very beginning, but all the way up to right before you analyze your sample, will take into account auto sampler variability. I've had cases where I'll run out of sample, or I'll have air in my system, and I'll inject half of my sample, and the peak area ratio is maintained, but my absolute signal of my analyte is less because I injected less sample. So there are a lot of issues that can be accounted for if you have an internal standard. And so here's an example of what you might normally see if you run a response curve without an internal standard. These are raw peak areas plotted on the y-axis versus concentration on the x-axis. And you'll notice the straight line linear regression through the points is supposed to be straight, but the points are like jiggered on either side of it, and they're not really in a straight line, right? But if I take that peak area of my analyte and I normalize it to the heavy peptide that's present in the sample, same exact data, I'm just normalizing it. Now those points went to the line, and now I have a better adherence and better linearity in that fit. And so that variability from sorry about that animation, that was weird. The variability that you see, or the distance away from that linear regression, is due to any kind of issues that are going on with my sample preparation, delivery to the instrument, detection at the instrument, but it's also happening to my internal standard, and so it's drawing it back to create this more linear response. Okay, so we've got differences between internal standard and external standard. So I always have been using internal standards. That means I take my standard, I spike it into my sample, it's analyzed at the same time. External standards are something that people use. For example, how many people have done the Bradford absorbance assay or the BCA assay for protein concentration? Right, show of hands. So what do? You take a standard, you dilute it, you generate a response curve, you get the absorbance measurements, and then you add the stuff to your unknown at the same time and get that absorbance measurement, right? And then you take the absorbance you get from your sample and you plot it on that curve. So that's an external standard. That's something that's not done with your sample together. It's done in a separate container, and you're going to the data plotted from that standard that was generated at a slightly different time or in a slightly different way, and you're extrapolating the information from your sample onto that plot. People do use external standards, and it's common, and there's nothing wrong with it, but they don't account for a lot of the variability we see in mass spectrometry. So it's, in my opinion, always a good option, if you can, to use an internal standard. And remember that if you are comparing against an external standard, the precision will obviously be higher, and there is not really a good accountability for the accuracy of your measurements. Okay? Any questions? So an example for these types of analyses of an external standard would be, if you don't have access to a heavy internal standard for your peptide, let's just say, but your peptide is detectable in plasma, and you're looking to detect it in plasma. If you dilute it in a different kind of plasma and generate a response curve of your peptide by diluting it with plasma that doesn't have it in it, so you're decreasing its concentration within a plasma background, that's a response curve, and you're able to generate a linear relationship with it, but it's not going to give you accurate information on a precise sample that you're measuring. You'll have to take whatever value you get from a separate sample and go to that plot and figure out the concentration. So does everyone see it's a little bit different? It requires multiple measurements, different samples. Okay. So the other issue about external standards is that it may not account for sample matrix differences. And again, this is another example using absorbance. So it says absorbance, but we see similar effects with mass spectrometry. And yesterday we showed examples of here, like the green plot, for example, would be similar to what we see with saturation. We would see the response of the peptide go up and then plateau because the detector can't continue processing the information it's getting because it's saturated. And so if you were to try to plot your data based on a theoretical calibration curve, that's fine, but if the actual response isn't linear, then you're not precisely and accurately calculating the amount of sample that you have, right? So these are just little issues that can happen with external curves or external standards. So for internal standard types, there are a variety of standards and a variety of places that you can add them. So if we go from the top middle down, we've got our workflow. You get your sample, you reduce, alkylate, and digest. You may or may not desalt, and then you go on to LC-MS analysis. So starting at the, I think I start at the top. Nope, I don't. Here we go. Starting at the top, you begin with proteins that go through that sample handling and digestion process. Heavy protein standards are not entirely common. They're sort of the gold standard of what everyone would love to have, but because it's difficult to make them, you usually have to go to some kind of expression system, and then those proteins have to be purified. It's not straightforward. So some people have gone to the global proteome standard, where you can either generate a proteome in heavy labeled media, so all of the proteins contain heavy lysine and arginine. This is fine if you can grow your sample in cell culture, but if you're working with plasma, you can't like stuff the C13 and 15 into your plasma proteins because they're already made. So another option for a global proteome standard, and there are a lot of papers, I think, from Dick Smith's lab, where they digest their sample with trypsin and then redigest it with heavy water, O18 water, and that O18 is incorporated into the peptide during the cleavage process, and I think you get a mass shift of 2 or 4, I forget what it is per peptide. And so that's a way to label it. Of course, when you're generating a standard, you need to qualify it, make sure that all of those masses are added in, and then figure out how much of it there is. So that's a reference to look up. If you don't have synthetic peptide standards that are heavy labeled for every analyte, you can use, let's back up. Extended peptide standards are something I'll go into. So this is something that you would add in to your sample before digestion that has, so it's the normal tryptic sequence, but it would have additional amino acids synthesized with it on either side of that tryptic cleavage site that are normal to the native protein sequence. And as trypsin comes in, it would see that and cleave those extended wings off. And so this would hopefully take into account the variability of the digestion step and reduction alkylation. And then synthetic peptide standards, this is the actual analyte you're going to measure. You spike it in pretty far down the workflow, and it doesn't take into account a lot of the variability preceding it, but it is pretty much the analyte that you're measuring. So one version, if you don't have the access for having a different isotopically labeled peptide for every analyte, this is something called the labeled reference peptide. So let's just say you have the Biognosis IRT peptides or the Pierce PRTC peptides. These are heavy peptides. They shouldn't interfere with if you're analyzing human samples. I think they're yeast, maybe they're E. coli. Does anyone know? I don't know. They're a different organism. Anyway, so they're heavy labeled. They shouldn't really interfere with what you're doing. And what you can do is add these to all your samples, not only for tracking retention time, but you can normalize other analyte signal in your sample to the closest peptide, or the addition of all of the peak areas from the peptides that you added in, these labeled reference peptides in your sample. So what you would do is just, again, it's a relative ratio. You would just do the peak area of your analyte divided by the peak area of one of these peptides, or the sum of all of them. And this way, it may take into account the variability of desalt if you add them prior to desalt. It may take into account the variability of autosampler injection. It may not take into account the variability of any ionization issues you have as your samples come off the column, but it is considerably less expensive. And if you use the same labeled reference peptides for everything, then you can sort of optimize on them once, feel comfortable with their stability, and then every new set of peptides you have to monitor, you've already checked out your internal standards. So what does it buy you? This is an example of a study that was done where the peak area of the light peptide is the same in all 3 squares, and then it is normalized in a different way in each plot. So on the left side, it's normalized to an exact internal standard that's heavy labeled, and what you see is the CV is calculated at about 1%, and the R square fit is lovely. In these cases, because the normalization is different, the axes on the Y are not comparable, so you can't really compare the slope. But the labeled reference peptide for that same light peptide analyte is just normalized to a different peptide eluting somewhere else in the gradient, and so its CV is calculated at 5%, but it's the same data, right? And then if no normalization is taken and it's just the raw peak area plotted versus concentration, you see the CV is 13%. So again, same exact peak areas from all the data from the light peptide, it's just being normalized different, and kind of like what happens with IRT. You see a bit of a scatter around that regression line, gets smaller and smaller as you normalize with an internal standard, an exact internal standard. So why synthetic peptides? One is that they're easily synthesized. We can add in heavy amino acids at any amino acid pretty much, as long as it's already available, which most of them are. They're reasonably inexpensive. You can spend a few $100s to get a pretty pure peptide that you can spike into over thousands of experiments, because they're usually prepared and sent to you in solid form or very concentrated. But again, they don't account for the variability from certain sample preparation parts, like the reduction alkylation of your sample and the desalt. You can add them when you do reduction alkylation prior to digestion, but there is no telling if, and you would have to check for this, if adding your enzyme with existing peptides in peptide form, if the enzyme might do anything to it. Theoretically, if it's a tryptic peptide, it shouldn't, but there could be other things going on. And you have to remember that when your peptide is being generated from the protein, it happens at some point, not at time 0. So if you add your heavy peptide in at time 0, it's got an offset from the internal standard is put in at time 0, and your actual light peptide is generated at t plus n, some other time that's a little bit later. So they aren't going through the same exact process at the same exact time. So can your synthetic peptide account for desalt variability if you add it before the desalt? Yes, it can. Some people don't like to add it before the desalt because you don't always get 100% recovery through that step. If you add it before the desalt and you lose a little signal compared to adding it after the desalt, that's actually okay, because it's taking into account your analyte signal that may be lost too. And then you're getting closer towards accurate quantitation. So this is an example that was done a few years ago, and this is from the Adana et al. Paper in 2009, where this was a study where in the first set of plots, the sample was digested plasma that had light peptides spiked in a response curve, and the heavy peptides spiked in fixed. And this is just two different instruments, two different sites looking at the same peptide. And they're pretty similar. You see the different colors or the different transitions, and there are 3 or 4 replicates at each concentration point. The second, experiment number 2, the light proteins were available for this peptide, and it was digested separately. The plasma was digested separately, and then a response curve was made from that digested protein. The idea there was to see how well does the protein digest in the absence of plasma, and then the last experiment was the protein was put into plasma and digested together. So we were looking at the effect of digesting with and without plasma to see if there were any plasma stuff that would cause problems with digestion. And what we found was, in fact, yes. So when in each case, each set of responses was normalized to a synthetic peptide, an internal standard peptide. And what we see here is that we get a very good linear response when we have a known amount of peptides, light and heavy. We mix them, spike them into plasma, it's already digested. You get really good response, really good linearity. This is what you should hope for. This is what you should like to see. Now when you start with a protein and digest it, and look at the same peptide, and spike that digested protein into digested plasma and add the heavy internal standard, now the slope is different. It's because we're not getting full recovery from the digestion of that protein. And that's due to digestion inefficiency. Not every peptide is released at 100%. And so if you have 1 mole of protein, ideally you would want to see 1 mole of every peptide it has released when it's digested. Quite often that is not the case, as this is an example. And then finally, when the light proteins were combined into plasma and then digested, it's even worse. So you can see that sample handling, how everything is treated definitely has an effect on the recovery. Recovery has an effect on accuracy of your method. And so the only way that you'd be able to compensate for this is by having a heavy internal protein standard. If you had that, and probably in both cases, these shallower responses would pop right back up to that diagonal regression line. So on the topic of peptide production and digestion, I recommend this paper. I have a PDF. If people want it, I can send it around. This guy, Chris Shuford, who used to work for David Muddyman, but now is a scientist at LabCorp, he did this study looking at the production rate of peptides during digestion and their decay rate. So what you may know is that every time you add a protease to plasma or your cell lysate or whatever, you're generating peptides, but they may not be stable. And while they're being generated, they either stay for a long time or they may decay over time. And you don't know until you measure that. And so this is just a good reference that it also looked at adding the internal standard at different points, pre-digestion, post-digestion, and so on. And that's where it's taking into account time 0, which is when you add the enzyme, and then at some other point in time in the method. So this gives you a good idea that you're not going to know what's happening to your protein and the peptides coming from it unless you go looking. So another option is what I mentioned to a couple of people in the room today. There's some people that like to use synthetic peptides that can be synthesized the same way with heavy labeled amino acids, and they have native amino acid additions on either side of the N terminus and C terminus where lysine would normally come in and cleave. And the idea here is that there's something for trypsin to go in and digest when you add your standard. So it's a good idea because you have to add them pre-digestion, but they're kind of a pain to work with. So has anybody worked with these before? Yeah. So the problem is that, remember when we had our little rules on synthetic or on peptides for targeting, we had that like 8 amino acids to 25 amino acids. Well, now add on like 6 to 10 more amino acids because that's how big they will be when they are synthesized in this form. So the bigger they are, the harder they are to synthesize. The likely less soluble they are because the more hydrophobic things you're adding onto it, the more likely the peptide's going to like crumple up and it may crash out of solution or stick to the tube walls, especially at low concentrations. And once you get it and it's synthesized and somebody gives it to you, there's literally nothing you can do with it until you digest it. And the best way to digest it is not all by itself in a tube, it's to put it in plasma matrix. And so now you're going to try to be optimizing something that you needed to do sample prep on, and now it's in a complex sample matrix. So they work, but they take a lot of front work in order to make them work really well. So the major benefits are that digestion variability tends to be minimized, and so when you normalize to this type of standard and it goes through the digestion process, you're reducing the perceived effect of digestion. So if a peptide with this winged form is released not in a 1-to-1 ratio with if it's similar to the protein, then you'll see more of a linear response with a slope of 1 compared to looking at an intact protein standard. And this is a bigger question that's not just about these extended or winged peptides, but how do you know their exact standard? You know, we trust whatever the vendor tells us. If you buy something, we hope that they're right. They're not always right. Most of the time, amino acid analysis is what's used to determine concentration. Does anyone know how amino acid analysis works? Okay, this is the very first thing I had to do at my job out of undergrad. I got a job at a biopharmaceutical company making proteins, and we had to generate a protein standard. We had to get its concentration. So you have to use positive displacement pipettes to work with a solution where your analyte is in there, and you pipette out specific volumes into glass vials. You add hydrochloric acid, 6 normal, and you hydrolyze overnight, adding a bunch of other things to label the amino acids that are generated from this process. And so you're hydrolyzing your protein or peptide into amino acids. Then you separate the amino acids on a very well characterized method that uses either fluorescence or UV detection. And then you look at the peak areas of each of your amino acids from your sample and compare it to an external standard of those amino acids, compare the peak areas. And it sounds pretty straightforward. If you digest a protein or a peptide into its components and you already know the sequence, you should be able to figure it out, right? Well, not every amino acid responds really well, and they don't all behave like the standards, and not everything hydrolyzes well. And if you messed up the pipetting, you are not going to get the right answer. And so this amino acid analysis process is like, you get a number plus or minus 30%-ish maybe. And so it's really difficult for us to absolutely quantify how much of a protein is present in a sample from a peptide or extended peptide standard. Even from a protein standard, it's very difficult for us to know for sure. I had always hoped, because amino acids can absorb and they can potentially fluoresce if they have certain amino acids present, that vendors would make peptides, heavy labeled peptides with like acid cleavable tags or protease cleavable tags, where you would know how much is in there by doing a UV measurement on a nanodrop or something. You would use like a microliter of your stock. It would be so awesome, but we're not there yet. But if you keep asking Pierce and Fisher and whoever else, maybe they will make it. So this is a study, again, there's an ASMS poster available where we evaluated this winged peptide experiment because we're like, oh, this is great. If we could make this work, it'd be awesome. We had a bunch of proteins that we had synthetic tryptic peptides available for. So those are the ones shown here. And then we had these winged peptides synthesized that were differently labeled than the tryptic peptide. And so you can see the base tryptic sequence is underlined, and then the heavy amino acids are shown in red, so that when we digested this and it was combined with the tryptic peptide, we would be able to get a different mass to charge, different product ions. And I'm like 90% sure we also had the protein available for each of these, or for most of these. And you can see the length, we called them winged peptides because of the wing things on the side. And we monitored 3 to 4 or 5 transitions per peptide. We also wanted to see if there was an effect, because these all had 6 amino acids on either side. We wanted to see if there was an effect on the number of amino acids on either side of that tryptic cleavage site, because trypsin might need to have more than one. If you've ever seen a miscleaved peptide, sometimes it's because they have redundant like KKK sequence or KR or something. So we wanted to see if that was what was causing it. The question was, is it a secondary structural influence, or is it a primary amino acid sequence influence on that digestion efficiency? Right, and we had to make some extra modifications. For example, this asparagine is not really an asparagine in the protein sequence, it's a glutamine, but glutamine, I believe, has the capacity to cyclize when it's at the N-terminus, and then we didn't know if this cyclization of a glutamine would affect the cleavage, and we were just grasping at straws a little bit. But in each of these 3 cases, we definitely had these proteins. And so the way we worked up the study was, we started with plasma, we spiked in the light proteins at a fixed concentration, and then we spiked in these winged peptides at the same concentration, or what we thought was the same concentration, did the digestion, added in our synthetic labeled peptides, the fully tryptic ones, at the same concentration. So we are really just looking for a 1-to-1-to-1, nothing fancy. We were well above our lower limit of quantitation, and we just wanted to look at these peak area ratios and see if they were consistent. In the process, we also, instead of just targeting, we did some data-dependent experiments to see if we saw any alternate forms of these peptides, because we were going to be targeting fully tryptic, but were we going to see some ragged ends, where trypsin doesn't fully cleave really well, and you might see some partial extensions or things that aren't fully digested? And so that's what we were doing with that. So these are the forms for the protein, the target tryptic base sequence. These are the forms from the protein that we observed. If you see more than one, that means we saw an alternate form. So here, that N-terminal lysine wasn't cleaved because it has an N-terminal arginine, so we see miscleavage. And we knew that these were from the protein because they were light. When we also observed form from the wing peptide that had internal heavy amino acids. And in some cases, ideally, The wing peptide is acting just like the light protein. We would see the exact same form. So if we saw an alternate form here, we would hope to see the same alternate form from the wing peptide. And in most cases, that's what we saw, with this one exception. In the wing peptide, it didn't cleave off that last 6 amino acid wing, which I could not tell you why. So here are just examples when we look at 2 peptides from the same protein, and we normalize the response of those peptides to the isotopically labeled synthetic fully tryptic standard. We get a ratio of peptide. These are 2 different peptides from the same protein. When we normalize them to a synthetic tryptic peptide, we get a ratio of 2 to 1, almost 7 to 1, 3 to 1 from these 2 different peptides from the same protein. Now, if we normalize them to the winged peptides, so this is the response of the protein itself normalized to the winged peptide. If the winged peptide is acting similarly to the protein, which is what we hope, we would expect this ratio to more approach a 1-to-1 response, where 1 mole of protein gives 1 mole of peptide, right? And so that's pretty much what we saw, except for an X and A7. And again, no idea what was happening there, other than no idea what was happening there. So it wasn't that we hit a home run on every single one, and probably maybe it's the exception that proves the rule, but still, when you can't get a full agreement on how something works, it makes it challenging to recommend it with a high level of confidence. So then we looked at the wing length versus recovery to see if there was an effect. And so these plots are showing the response of the peptide generated from the protein in blue versus amino acid wing length, and then the average recovery of the peptide generated from that winged standard in red. And these are both normalized to the internal synthetic fully tryptic standard. And so what we would ideally see is if it needs a longer amino acid wing length, then we would see higher recovery at the longer amino acid wing length end. And we kind of see that effect, but it's not consistent from peptide to peptide. And so this made us kind of wonder if it was a very local amino acid sequence effect, like perhaps the individual amino acids and where they are located in proximity to that cleavage site actually control how quickly trypsin can go in and do its job to cleave and generate a fully cleaved peptide. So a lot of like, oh, I'm not sure, you could interpret it this way. So if you do decide to proceed with winged peptides, just be careful. Make sure you have in your mind all the potential experiments you could do and think about the potential outcomes so that you have a good way to interpret them. Okay, heavy protein standards. So it's the best way to get the closest to accuracy you can without adding too much to your sample. If you do SILAC, you're adding an entire proteome to your sample and effectively diluting it by a lot. So that's why a lot of people prefer to go to heavy peptides and heavy proteins, an individual case-by-case scenario, than adding an entire additional proteome. So these can be grown on heavy media that has an N15 source and purified. That's how the ones that we used in the CPTAC study were made. I believe they were expressed in coli. And you can add in heavy arginine and lysine. This is the same way that proteomes are labeled in SILAC. And you may be able to, these may be difficult to produce or purchase. And the question is, once you have the heavy protein, how do you ensure it is the same exact sequence as your light protein, right? So you have to do first experiments to try to combine them and see if you're seeing the same exact peptides. If you have sequence variants, you have to figure that out. So it does take time to do some characterization of that. Proteins, however, going back to amino acid analysis, I think they're better suited for amino acid analysis because when you hydrolyze a protein, you get many, many, many of each amino acid because you've got very large proteins. Peptides, you might get 1 or 2 of a particular amino acid, and that's just not giving you as good quality data as you would. But with proteins, I think overall you tend to get closer concentrations of your stocks when you do this procedure. It's not fun though, let me tell you. And I exploded an acetonitrile bottle there too because I put a waste bottle next to a vacuum thing. And health and safety was called. It was my 3rd week on the job. I thought I was getting fired. Good times. So this is an example from that CPTAC study, and we'll go into it with the data file I had asked you to to open up. And in Skyline, so you can have multiple labeled forms of the same peptide. And here, so red is usually light, blue is usually heavy, and then the next color, if you have a third form, is brown. Like, could it be something prettier, Brendan? I mean, like brown, anyway. And so in Skyline, you can normalize by one internal standard or another internal standard. So when you take the C13N15 peptide and you normalize that response to the synthetic tryptic peptide, and you plot its response, and this is shown on a log scale so we can see it clearly, you see that it's falling below the theoretical line and it's obviously plateauing down at the bottom. And if we pick one of those data points and we calculate how much is actually present based on the light to heavy peak area ratio and how much we spiked, this is a 1.3 femtomole concentration point. We're calculating it's actually present at about 50% recovery from the light protein that was in the sample. If we normalize instead that same peak area to the internal standard protein that was spiked in and calculate the recovery based on that spike, we're at about 100. And so again, this is exactly what we're looking for. We just want to show that we're getting accurate quantitation as well as precise. So again, as I mentioned earlier, SILAC or global heavy standards. If it's SILAC and you're able to grow cells in culture, you wind up growing the same sample, it's just heavy. All peptides should be present in the heavy form, but the standard may be difficult to quantify, as I had mentioned, because it's so complex. Results may not be absolute unless you can figure out how much of a peptide you have in your internal standard, is how much is there. You can't compare it to your light form and then say anything more than a fold change. And so that may be useful, but it's definitely not absolute. Post-translationally modified peptides may also be accounted for, whereas in working with synthetic peptide standards, they usually cannot be unless they've been synthesized that way, which is challenging. Phosphopeptides, we know they can be made, but others may be a little more difficult. And then if you run out of your global standard, you have to make more, and then you have to requalify it. So that's also another challenge. And this goes for any standard. If you're running low on one, you just want to make sure that you generate more with enough time to overlap and make sure that they give similar or parallel performances before you completely convert over to the other one. And then as I mentioned before, O18 labeling during digestion may be another option, but you need heavy water, and that's not cheap. It comes in ampules, so when you're going to do it, you have to be like, nobody talk to me. I need to make sure I'm doing everything right and I don't drop anything, and so it just takes a lot of care. So you've chosen your standards. Now what do? Construct a curve. So there are things that you need to think about, like what spike level will I use of my heavy? How many points do I need to make for a response curve or a calibration curve? Then you have to acquire the data, then you have to analyze the data, and then you may actually have to repeat all that process because you didn't do it right the first time. And that makes you sad. Maybe not. So why run a calibration curve? So there are some methods of calculating LOD that rely on it, and this is how I was trained. I went to the University of Florida for grad school, and James Weinfordner, who's a professor who retired a couple of years ago, and he's like the pinnacle analytical chemist. And if any of you have taken analytical chemistry and used the Scoogin-Leary textbook, he is in that section that talks about limit of detection. Mostly it's for spectrophotometry, because that was his area, but a lot of the same rules apply. And so he reported an equation that was very simple and easy for someone like me to do in Excel with my data. So that's what I usually use, and it takes into account the signal of your blank sample, the standard deviation of your blank sample, and the slope of your curve. And you take all of that, it gives you a concentration, and you're good to go. So it requires you generating a curve. So do you need to demonstrate linear range? That's one question. As we had mentioned a couple of times, I don't like to really dirty up my system. if I don't need to show the upper limit of linearity, so I often don't go very high, but there are some cases where people like, how much can I load and what's the carryover effect? So it is worthwhile if you have the time. And because the point is, at the end of your method, you don't want someone to be like, you didn't do this and I have questions about that. Go, crap. Then you have to go back and do it all over again, because repeating portions of a response curve are not very useful. So what is validation? This is the next steps or things. Yep, let me just make sure. So we kind of inadvertently validated our CPTAC method because we wanted to set it up correctly to make sure that it was top quality experimental design. And looking back in hindsight, I now see like, oh, we actually kind of validated some of these things. Now, validation, I use the term loosely, and I'm not using it correctly. Validation, again, as I had said before, in like pharmaceutical companies, has a very particular meaning. But here it's defined as the process used to confirm that the analytical procedure employed for a specific test is suitable for its intended use, right? So you want to set up a method that detects your analyte with a certain level of precision, a certain level of accuracy, and a certain level of ruggedness and robustness. So Lindsay covered repeatability and some similar aspects like that this morning, and that is completely part of the validation process. So here what we did with our study 9 study is we looked at sensitivity, linearity, reproducibility, and so on. We included blinded samples so we could get accuracy. Robustness was evaluated by looking lab to lab. Could people perform with the same method, the same protocol, the same sample kits, and get the same answers? And then specificity is another aspect of true assay validation, where it just makes sure that you are getting the proper answer without false positives and false negatives. So before you begin doing your response curve, you need to know how much of your internal standard you want to spike in. And I've gotten this question from a few different people already. How much do I put in? A lot of it relies on the technology that you're using, so you want to make sure you have an idea. Like, I know off the top of my head most state-of-the-art triple quads that have been made in the last 5 years up to present. You need a minimum of low femtomole to see a pretty good signal using nanoflow with a 1 microgram sample load. For high flow, I might do 10 to 20 times that injection amount, which means 10 to 20 femtomoles per 10 to 20 micrograms, right? So just knowing that, you can begin to set up a quick experiment to evaluate how much heavy to spike. So here we looked at 10, 5, 1, and 500 attomole on column just to see what we could see. And ultimately, we conducted this experiment. So blue is heavy, red is light, red didn't have any spiked in it. This is in our sample matrix. So we also wanted to look for potential interferences, and the idea is we see a different response. If you want to try to see this view in Skyline, you can do synchronized zooming, and you click on the most intense one, and I just wiggle it around a little bit, and then everything else has the same y-axis and the same x-axis. So that makes it nice and easy to compare. Or you could look at the peak area plot. I plotted it in log, so you could, otherwise the response is so sharp you wouldn't be able to see some of these things. Yes?
You also have to unlock the y-axis. By default, the y-axis.
That's right, I forget. Okay.
The right click with auto scale y-axis.
So ultimately, we had this conducted at a few different sites on different platforms, an old one and a newer instrument, and decided that 10 femtomole was the spike level we were going to pursue. So then next, what was the concentration range for the curve? You know, what do you want to shoot for? Do you need to demonstrate orders of magnitude? You know, a lot of this really depends on your biological assay. If you have no idea, then you do something wide and broad, like we did for the study 9 method. But here, this just goes through a little bit where for limit of detection, so this is something where how low should you spike your samples. Again, current state-of-the-art triples can detect less than 100 attomoles on column. And some of them, depending on the sample matrix, can go down to 10s to 1. and it's about 10 to 20 times higher at higher flow rates. So if you know this, then you can probably set up a low end and a high end, and then what I like to do is convert to the log scale so that the points are equally spaced. Although sometimes I like to cluster them up around where my LOD is, LOQ. And the idea there is you want to keep going to lower and lower concentration so that, at least what I'm comfortable doing, you keep going lower and lower to see your signal decrease, right? So you don't see, and then you go one step lower. So you don't just stop at the point where you're like, oh, I still see it, so that's my LOQ, or that's my LOD. I've seen people do this where they'll do a response curve and they'll do 4 points, and their lowest point they can still see their analyte. They're like, that's my LOD. Like, but did you go lower? No. I can still see it. They're like, but that's not your LOD. You don't know what your LOD is. You can keep going lower. You keep going until you get 1 point more and you can't see it anymore. Then you can at least have a ballpark of your LOD. And these are just some quick and dirty ways of assessing LOQ and LOD. And again, these are very quick and dirty if you don't even want to go into Excel. A lot of people look at the replicate peak areas, and if their RSD is around, well, I guess 15%, signal to noise is greater than 20, that's the LOQ. If signal to noise is 3 or greater, then that's your LOD. And your percent RSD should be low as long as you're above your limit of quantitation. And again, these are topics that Cyril and Mani will be going into later. And then keeping in mind, so I can't say for sure I know of any instrument that fully detects 1 attomole of a peptide on column in 1 microgram plasma, but this is where it would be in the realm of plasma concentration, and nanograms per mil is pretty good. This is without any enrichment. So that just gives you a ballpark of what the moles are that are detected by the mass spectrometer versus what the concentration of the protein would be in the plasma, which are kind of 2 different languages, right? It's like what we detect versus what the clinicians speak. But ultimately, these units are what fund us, right? The biology and the cancer and all that. So for CPTAC study 9, these are the concentrations that we used from 10 attomole on column, and this is for the protein level spike, the light protein, all the way up to 100 femtomole per microliter, and the background sample matrix was 0.5 microgram per microliter. Our internal standard spike of the synthetic peptide was 10 femtomole per microliter, and the protein heavy internal protein spike was a little bit higher because we knew we weren't going to get 100% digestion efficiency, and we wanted to make sure we had a chance of seeing it. So that's why we added more of that. And these are just examples of looking at the response of a peptide on the linear scale and on the log scale. Ultimately, the limit of detection for this particular experiment with the protein level spikes was calculated for each of the sites that was able to survive the study. You'll notice there's less than 10 here. And if we look at the limits of detection where those LODs were calculated when we normalized to the synthetic tryptic internal standard, they're all less than 200 attomoles per microliter. And if we use the protein internal standards to normalize and then calculate the LODs, they're all higher than that. So the LODs are higher because the proteins don't always digest. And if they don't always digest to 100%, you're not going to detect them. And again, getting to better accuracy in this. So here's data from the blinded samples where, again, these are the different sites that looked at that analyzed the sample kits. And then the reported or calculated concentration based on the peak area ratio, where on the left we use the C13 N15 peptide to normalize, and on the right we use the N15 proteins. And what you see is that the theoretical accurate, proper concentration is shown with these horizontal lines going across, and they're color-coded where the darkest is 75 and the lightest is 4 femtomole per microliter. So that's how much was spiked into the sample in light protein. And then when we normalize with the C13 and 15 standards, we don't get full recovery because we're comparing it to a fully synthetic tryptic peptide, and there's no way we're getting 100% recovery. But when we normalize it to the N15 protein standard, because that N15 protein standard didn't digest just as poorly as the light, then we're able to calculate with better accuracy and better precision because it accounts for most of the variability for all of those peptides across all of those samples. So this is the reason. This is one of my favorite figures because, again, it's the same exact data. It's just using two different standards to normalize it. So how do you qualify your standard? This is really important because kind of like what Lindsay was talking about before, where how you analyze your data. If you use garbage analysis, you get garbage output. If you have a garbage standard, you have garbage data. And so how do you know its concentration? Honestly, I still don't know if there's a really good way. Like, we have amino acid analysis. It's like saying, I don't know, like that one bumpy road with thorns and stuff is the only one out of town, but at least we have it, right? It's just, it's our only option. Do you know if it's stable in storage? So this goes back to the repeatability type of experiments where, again, if you're going to spike in 10 femtomol into your sample, make sure that your standard has not degraded and you're actually spiking in 1. And how do you know if it's stable in storage, in the freezer, on the benchtop, in the auto sampler? So these are all things that you have to pay attention to because you're using that standard to state how much is in every other sample you're measuring. So test and test and test again. Refer to Lindsay's talk. Okay, so the rest of this section is a lot of hands-on, and we have until 3, and then from 3:30 until 4. So we have about a total of an hour, not including the break, to go through this. So what I've done is I've set up sort of screen capture views of what I'd like for you to do live on your computers, and so you can always come back to this as a reference. But first, if you could open up the file that begins with study 9.2, site 56B, the post DS one. When you open it up, hopefully it should look something like this. We good? We good? It has more to the file name than just post DS. I just ran out of room. Everyone okay if I go to the next step? Okay. Oh yeah, if you see the thing for a spectral library, just skip it and open the file anyway. You don't need it for this. By the time you're at calibration curves, if you still have to look at spectral libraries, I don't know, for this kind of work. So the next step after you have it open would be go to settings and then peptide settings and click on the quantification tab that's shown here and set the parameters as shown. So some of you, I may have done this before, and if I did, it might be saved in the Skyline settings, but some of you might have to do this. And so you want the regression fit to be linear. The normalization method, do you see ratio to heavy and ratio to 15N? You see both? Okay. So for now, we'll just do ratio to heavy, and then later you can go and change it if you want to look at the other way. I don't have regression weighting, MS level, leave it at all. And then the units is just for your own reference. I have it as femtomoles per microliter. All right, and then the idea here is I've given you a Skyline file that was generated before Skyline was capable of doing calibration curves. And so you have the data, it's fully integrated, and now we're going to set up the calibration curve so you can interpret that data. In order to do that, you may have to go into document settings, so settings, document settings, click on the annotations tab, and make sure that you've got sample group, concentration, and IS spike checked. And again, some of you, I did this before, and hit OK at the bottom, don't click add, hit OK. And so now, if you go to view results grid, and what is it? Do you have to be on a peptide, or you might have to be clicked on a peptide? You should see this, and the point being is you should see these 3 columns that are empty. Is anybody having problems?
That was a little too fast for me to keep up.
Okay, I can go back. Oh, document settings, it should be settings, document settings. Yeah, sample group, concentration, and total standard. Right? How many people are still working on it? Not sure. All right, I will go on to the next step. Are you ready? Okay, thank you. So if you go to View, Results Grid, how many people do not see this view? Because I know sometimes it depends if you have selected a peptide or a precursor, the view may be a little different. You don't see it? So you should see, what you may be able to do is, you see this little drop down views thingy? If you drop down it, there should be a view called peptide quantitation. And you may be, you may need to be clicked on a peptide, the peptide sequence. So just, you pick the first one. You got it, okay. Great. Yes, so this results grid view will change if you are on a peptide sequence or its master charge precursor or a product ion. So for this, where we are, just make sure you have that first peptide selected, and then you should see everything the same as here. Okay. Everybody good? All right. So now we need to populate the sample group. The sample group is how you are telling the software which sample we have replicates of that can be grouped together to generate average peak area or coefficient of variation. And it's an alphanumeric string, so if you put in concentration all in the same units, like 0.1 and 1.0 and 10, it sorts them not in a numerical order. So I always go with text. And so I usually will label them A through whatever. Now, please make sure you note the QC that I have in the middle. That is not a standard. That's not a calibration curve standard. And so you can label it as QC. And what I would also like to show you, if I may, is when you have this open, View Results Grids, I'm going to show you some tricks. If you click and drag after you populate that first A through J, and then Control C, copy it, and then go down to the next line, you can paste. Control V, and it'll paste below in that next set. And then you can do it again and again. Are you guys seeing how, is that working? Okay. What's that? This is the post DS study 9.21. Okay, and then let's go back to here. Okay. Yeah, if just open the file. If it asks you about a spectral library, don't browse or go looking for it. Just open the file. Okay, so the next step after, is everybody caught up and you've populated that whole sample group section with A, B, C, D, E, F, G, H, Q, C, I, J, all the way down? I think also there is a system suitability run that inadvertently got tucked into this file down at the bottom. So if you, no, I guess not. Never mind.
And how do you edit the table? So my table doesn't have the same columns
Okay.
that you have.
So for that, go to Settings, Document Settings. Do you have those, at least those three, sample group concentration?
Mhm.
Okay, so those are the most important ones that you need right now?
Okay.
What other, are you missing ones that you?
Sample time.
Sample type. Do you have for views, if you click on this drop down, you see where I am?
Mhm.
Is peptide quantitation an option for you? Okay. So-
Peptide results, custom or managed.
Are you clicked on a peptide or a precursor? Peptide, okay. Brendan, I may need your help. Eh,
No, views. And then customized.
it should be a default view though. That's, I would like for it to just be there because it should. So some people are not observing the sample type column, and when they click on views, they don't have peptide quantitation.
But yeah, get on results grid. And then go to document grid.
Okay.
Yeah, we should have sample type right next to the rest.
Does everyone have that now?
Where'd you get that? Sorry.
It's under view at the top,
Yep.
document grid. And then under that, you go view replicates. Okay. No, but, okay, yeah. So then, what you wanna make, this is where, and Monty, we'll have to talk later about quasar and which column it needs, because the response curves use analyte concentration and quasar uses concentration, but they're the same value, so if we can, okay. So for this view here, if you would humor me, if you take your entire column, whichever column you have that has the concentration values in it, I'm not sure if it's, you don't have anything.
It's not there, it's not the replicates. If we're on document grid, these columns are
Yep.
Are you clicked on a peptide or a precursor?
peptide.
Okay. And under views, you're under replicates? Brendan? They're not seeing these options under document view replicates.
The sample group and concentration ion spike. All this thing?
Oh, I thought you said that these concentrations. Well,
No,
also
no,
these
no.
aren't built into the sample method.
Yeah, no, you have to do that.
Okay.
Yeah,
That
this-
was not, I guess we didn't.
So this file, when you open it, because this file was made before you could do calibrations in Skyline, it won't have any of this information. So we are walking through the process of populating this table to get you where you need it to be.
So we have to type in all the concentrations.
You don't, well, yes and no. So hold on one second.
Oh, okay.
I have this. So has everybody at least gotten through populating the sample group column? All the way down. All right, good. So next is you should, and you will probably look at the document grid, don't use the results grid. You wanna, for the internal standard spike, if you type in 10 in the first column, click and drag it, and then right click, copy down. So the click and drag part is a nice feature. I can't hear you, sorry. You type it in, and then if you type it in A through J, you can copy this and paste it down in the section below. So this is all like selectable, copy pastable. Is it? Is anyone else stuck? Show of hands. We'll come around. Okay.
Where you stuck?
Yeah, go to view documents. Right, yeah, do everything, fill out everything in your documents grid, and I will continue walking you through how to fill out some additional information like concentration. So, here we go. So has everyone filled in the internal standards by column with 10? Okay, good.
What I received samples that.
We'll get to you, hang on. I'll get to you in a second and we'll fix that, because that's easy enough now. So, I sent this last night. This is an Excel file that has the decoder key to our sample type, our name, which is A through J, and then the analyte spike. So, open up that Excel file. We good? All right, because I'm going to show you an easy way rather than typing it in lots and lots of times. So what I'd like for you to do is once you open it up, you should see sample type, analyte spike, right? These two columns. Insert a line in Excel under the H. Put a QC in that empty row, and then keep the analyte spike cell empty. Okay? Okay. So now when you've done that in Excel, if you highlight that 0 through 100, just that whole range, and copy it, then you can put your cursor on that first analyte concentration cell and hit paste. And then that whole column should come right in and match up with your, every sample A should have an A and a 0, B should have B and 0.01, and you can just keep copying this down for the 4 iterations of the curves. You got it? Because you look like you're happy, so that's good. So you see the system suitability in there? That was my mistake. I think I show, if you go down and just keep that value for analyte concentration blank for that system suitability, because that shouldn't be in there, and then move all the others down. So the concentrations for analyte concentration should match up with these ABCDs and so on.
So there was one in the document grid then, is that
Yeah, so just keep that analyte concentration blank and shift the guys below it down. You should be able to highlight them and move them down.
That's what you just said.
That's okay. You're gonna find out. As a parent, you just repeat yourself a lot. Okay. Is anyone majorly confused? Do you need help? Okay. What's your question? Populate the rows the same exact way. So when you have one column done, copy paste it. The QC, just leave it blank, as I don't remember it. I think it's in the, that's not relevant for the curve right now. So All right, any questions? Okay. Are you in results grid or documents grid? Go to documents grid. View, documents grid. So then go to under document grid and see reference. There you go.
Yeah.
Okay, so I had said it a little earlier, but maybe not with emphasis. There are, not here, because I have a results grid. In your documents grid view, there are 2 columns that refer to concentration. It is redundant, but they are both necessary for what I want to show you today. So analyte concentration is required for plotting these in the calibration curve, and then the concentration column is required for processing the data in quasar. Okay? So this should pretty much be what you have. Is anyone stuck? Yes.
I don't have the analyte concentration column. I have the other one, but I don't have this column here.
Okay.
I missed one step maybe.
Yeah, so go to view document grid and then click on replicates.
Okay.
Did it show up?
Oh. It showed up.
You're welcome. Okay. So is everybody's table populated so far? Okay. So now with as long as there's everyone's okay. So now we want to do the sample type, and I'm gonna again just take you through the walkthrough section. Please use the I guess use documents grid instead of results. I didn't know. What you want to do next is I didn't have it in here. We need to identify the sample type in order for it to realize whether it's a standard or a blank or something that needs to be quantified. So the way that you do that, I'll try to show you the easy shortcut way, is A is a blank, and it's always a blank. So you just click that drop down and select blank, and then B, you'll click the drop down and click standard. And then if you have that highlighted, don't click anywhere else yet. Hold the shift button down and click on the 8, like through H, and then copy down, fill down. That will go bloop, and then you're good. The QC one, it's a quality control. I know that it's heavy and light peptides spiked in and no sample matrix, so the data is not meant for the curve here. So I just have it as quality control, and for now we're going to ignore it. And then for I and J, make sure they're standard. Once you have that first series, the A through J labeled, you just have to do it once, then hit Control C. Oops, hold on. Control C. And then when you click on the A2, you can Control V, and it'll paste into the column. Now you can do that all the way down, except for if that system suitability run is in there. Just make sure that that's put in as an unknown. So again, I'm sorry that's in there. It shouldn't be. Okay.
And what do you want the system suitability to be labeled as?
Unknown. Does everybody have them all labeled? Okay. So now, what I do is I'll take this and I'll anchor it back down into this guy, however it happens. And then if you don't already have a calibration curve window, you can go to View, Calibration Curve. You should see something like this. Oh, please. I hope you do. All right. See, heads nodding. Good. See it? Yeah. I don't blame you. All right. So what's nice about this, for those of you that have not gotten here yet, I'm going to keep moving a little bit because we're close to a break. We're about 10 minutes from a break, and I just want to get a little bit more. We'll have time after the break to catch anybody up and to answer specific questions that people might have about more things you can do. But for right now, we have a response curve. I have the first peptide clicked. If I go to any other peptide, it will change the response curve because the peptide response changes, right? So that makes sense and pretty good. And so if you go edit, collapse all precursors, edit, collapse all peptides, then you can just simply use the arrow button and just look at all of your curves as you walk down. So it's kind of interesting to explore that way. The other thing you can do in the calibration curve section is if you right click and you click on log plot. Again, this is nice because when you're not on log plot, there are actually 9 points in this curve, but you can only see 1, 2, 3, 4. You can click and zoom down at this low end, and then you can see them. And you can see that they don't very well adhere to that linear part of the curve. Or if you go to log plot and then zoom out, undo zoom, let's not do that. There you go. Then you can see whether it's a straight linear shot or if it does wind up plateauing at the bottom. If you also right click, show sample types allows you to toggle the display on and off for unknowns, for quality controls, for blanks. So if I clicked and had blanks in there, they would show up probably way down here somewhere. A blank is shown in the legend, right? So this file is, once you have this calibration set up, you can just save this file wherever it is. Just click the save button and explore it. Now, the other thing, I don't think there's a way to do this. If you want to look at normalizing against the N15 labeled protein, you have to change which internal standard is being used to normalize. So here, you would go to peptide settings, quantification. Now it would be ratio to N15, and you would hit OK. And now you have to change your internal standard concentration. So Brendan, here's a question for you. If you have two different internal standards and you want to look at it normalized to one and then normalized to the other, do you need to have two different saved Skyline files to do that?
That's not anything we were, not anything that we told us they needed, so.
Okay.
Nothing but what we have.
Okay.
Do you need to read this, send it in an email?
I think so. Let's do it. So, after doing this cell population in the documents grid, if you want to look at and see how the curves change when you normalize to the internal standards, you do what I did, which is settings, peptide settings, change the quantification to ratio to 15N, hit OK. Now you need to change the internal standard spike, because you may remember from the Excel file, the internal standard spike for the protein is 25, not 10.
What sounded
I thought it did. I can't keep track of what Skyline does. It doesn't talk to me. I mean, for record keeping, if I'm normalizing to the 15N, I would want to know, 25, sorry.
So I think, actually now that yeah, I think, so if you want internal standard concentration, I'd say, that's not helpful. I think I have like the way
Well, that's okay, because we're gonna be doing Quasar too.
Yeah, I think you would go, so if you want to do it for Skyline, where we would find it, you would go to use peptide quantification view, the document, peptide quantification, and then where it says internal standard concentration, it's set that.
Okay. Oh, that's nice. Okay. Where would it use that if it was calculating an unknown, but in any other case? Okay.
If you have a linear,
How do you, in peptide quantitation, how would you be able to define the, I see. So it would be considered a surrogate standard? Because if the choices are, right, IRTQC global or surrogate, it's a surrogate. Leave it.
that peptide itself is not.
The peptide, okay, gotcha.
When I have an internal standard for this peptide, I make the norm.
Okay. So then,
You don't have to fill out that normalization method either, that's only if you didn't have it already set, like that's like specific. If you want to normalize that peptide differently from everything else.
okay, so what does this internal standard concentration do?
That would be if you were gonna use that to determine your one.
For unknowns or for the curve? Okay, all right. So if we go, okay, so if we change the internal standard spike to 25 and we went to settings, peptide settings, and change the quantification normalization method to ratio to 15N, then we can look at the calibration curve and see how it changed, if it changed at all. And so probably the slope would change. It's actually pretty good. So what I'm looking at, I haven't looked at these in a while, this is what is a nice thing to see. So remember how yesterday and today I showed you what an ideal quantitation experiment would look like with the response curve is y equals b. And so here, if we look at the slope, it's 0.99, which is pretty close to 1. And the y-intercept is 0.007, negative 0.007. So that's pretty awesome, right? And if we were to, so if I take and I save this Skyline file now as, because it used different normalization methods and I don't want to have to change back and forth my spike level between the N15 protein and the synthetic. Instead, because it's me and I have way too many Skyline documents open at any given time, I would just save it with a new file name. So if you didn't save the other one, now you'd have to go back and put the synthetic internal standard peptide settings in. But point being is, the slope and the y-intercept that you get from using the synthetic internal standards, the peptide, the tryptic ones, is probably not going to be so close to slope of 1 and a y-intercept of 0. So that was fun to see that. Okay. So I think we're at 3. We've got a 30-minute break at 3:30, from 3:30 to 4. What I would like to do is, I have another data set if people want to check out trying to do another calibration curve. It has a lot more peptides than this, but only 15 of them have internal standards and the others are not really in a response curve, so I guess it's not that exciting. But anyway, if you're interested, I can walk you through how to set that up, and I think those should be on the flash drive. Or in the afternoon, in the next half hour after break, before we hear about LOD, LOQ, if there are things that you haven't yet done and you'd like to do, like paste a peptide list into Skyline, we can do that. If you want to paste a peptide into Skyline and make sure the spectral library matches, we could do that. So when we come back, or now if you have suggestions, let me know, and we can begin setting that up for after the break. All right? Great. So in 30 minutes, we will all see you back here.